40% of your body by weight is just one thing. It's,
Hmm.
it's not bone. It's not your organs. It's not even water, really. It's skeletal muscle.
That sounds about right. Yeah, it's the biggest single component of our mass.
And I had this realization, looking through the stack of source material you sent over for this deep dive, we're talking about Chapter 6, Contraction of Skeletal Muscle, that despite it making up almost half of who I am, I have basically no idea how it actually works.
You are definitely not alone in that. It's one of those things we take completely for granted.
No, but I mean, really.
Yeah.
I understand the basics. If I lift a heavy box, my arm goes up, I get that. If I go for a run, my legs get tired. But the mechanism, how a thought, just an electrical flicker in my brain, turns into actual physical force that can lift my coffee cup. It's a total black box.
It's a black box for almost everyone. We tend to think of muscles as meat, just these slabs of tissue that get bigger if you eat protein and lift weights, and small if you sit on the couch.
Right.
But if you actually zoom in, and I mean really, really zoom in, down to the molecular level, you find something that looks a lot less like a steak and a lot more like a high-performance biological engine.
That is exactly the vibe I got from the reading. It didn't feel like biology. It felt like I was reading a mechanical engineering manual for some kind of, I don't know, a futuristic robotic arm.
It essentially is a robotic arm. It's just made of organic material. And it solves engineering problems that our best roboticists are still banging their heads against today.
Like what, for instance?
Oh, everything. Efficiency, leverage, fuel storage and distribution, self-repair. I mean, just think about it. Imagine a car engine that could rebuild its own pistons just because you drove it hard on the highway for a few days.
Right. That's what hypertrophy is.
That's exactly what muscle does. It's an adaptive, self-repairing, incredibly sophisticated machine.
So that's our mission for today. We are going to open up the hood on this black box. We're going to strip it down piece by piece. We're going from the bicep you can see in the mirror all the way down to the individual atoms that are literally pulling against each other to let you hold your phone right now.
We're going to look at the architecture, the spark that starts the engine, the fuel it burns, and even the grim reality of what happens when that engine shuts down for good.
It's a bit of a forensic examination of physiology.
I like that. Let's dive in.
Okay, Section 1, the architecture, or as the notes put it, the architecture of strength. The first thing that it floored me was the structure. It's not just a blob.
Not at all. It's profoundly, deeply organized. The best analogy, and the source material uses this one too, is a set of Russian nesting dolls.
I love that image. It makes so much sense.
Yeah.
So walk me through the dolls. We start with the biggest one, the one on the outside.
The big doll is the whole muscle itself. Let's use your bicep as an example, that big visible mass in your upper arm. It's wrapped in a tough sheath of connective tissue called the epimysium.
And at the ends of that muscle, the tissue doesn't just stop. It transforms.
Exactly. It fuses. That's the key word. It fuses with the tendon fibers. It's not like the muscle is glued onto the tendon as a separate piece.
So it's not two things stuck together.
No. The outer skin of the muscle cells themselves actually blends into the collagen fibers of the tendon, which then bundle together and weave themselves directly into the outer layer of the bone, the periosteum.
So it's a single, continuous mechanical chain. There are no weak links.
There can't be. If it wasn't a continuous structure, the first time you tried to lift something really heavy, you'd just rip your muscle right off the bone. The structural integrity has to be absolute, from the very center of the muscle all the way to its anchor point on your skeleton.
Okay, so that's the outer doll. Crack that open. What's the next layer inside?
Inside, you find bundles. They're called fasciculi.
Fasciculi. That's one of those words you see in textbooks.
It's just a fancy word for bundles of fibers. And you've seen these if you've ever had slow-cooked pulled pork or brisket.
I was just going to say, is this pulled pork? The stringy bits.
It is literally pulled pork. Those individual strings of meat that you can pull apart, each one of those is a fasciculus. It's a bundle of muscle cells, all wrapped in their own little connective tissue blanket, the paramysium.
Okay, so I take one of those strings, one fasciculus, and I pull it out from the muscle. I crack that doll open. What's inside?
Now you're getting down to the really interesting part. You're looking at the muscle fiber, and this is a really important distinction. The muscle fiber isn't just a strand of material, it is the cell.
It's a single cell, the whole string.
One single cell. Yeah.
But wait a minute, the notes say these things can be huge, like centimeters long.
They are absolutely massive on a cellular scale. In most of our skeletal muscles, that one single cell extends the entire length of the muscle.
So Let's take a long muscle. What's the longest one? The sartorius in the thigh.
That's a great example.
That can be, what, a foot and a half long in a tall person?
It can, yes.
And you're telling me that's a single cell? One cell that's a foot and a half long.
A single continuous cell. That's right.
That just breaks my brain a little bit. I'm used to thinking of cells as these microscopic blobs. You need a high-powered microscope to even see.
Not in muscle. These are the titans of the cellular world. They can be anywhere from 10 to 80 micrometers in diameter, which is actually just on the edge of being visible to the naked eye if you have good lighting and you look closely.
Wow.
And because they are so enormous, they have a special adaptation. They're multinucleated.
Multiple nuclei.
Hundreds of them sometimes. They're scattered all along the inside of the cell, just beneath the membrane. Think of it like a factory that's so long it needs 100 different managers, each one responsible for their own section.
That is a great way to put it. A single nucleus couldn't possibly create enough protein and manage all the metabolic needs to maintain a cell that size.
It would be impossible. So you have all these nuclei distributed throughout the fiber, and this entire factory, this giant cell, has a wall around it called the sarcolemma.
The sarcolemma.
That's the cell's skin. But it's not just a flimsy membrane. It's the plasma membrane, reinforced with an outer coat of polysaccharide material and collagen fibrils. It has to be tough. It has to withstand the incredible mechanical stress of contraction without tearing.
Okay, so we're inside the cell now. We're inside the factory. What's the actual machinery inside?
We open the next and final doll. Inside the muscle fiber, floating in the cytoplasm, which in muscle cells we call the sarcoplasm, are the myofibrils.
Myofibrils. Right. We're getting into the tongue twisters now.
I know. Just think myo means muscle and fibrils means little fibers. These are the tiny threads that run the entire length of the cell in parallel. Each muscle fiber, each cell contains hundreds, sometimes thousands of these myofibrils packed together.
And this is where we start to see the stripes, right? Because when I look at diagrams of muscle, it always looks striated. It's got that barcode pattern.
Exactly right. That characteristic striation of skeletal muscle comes from the myofibrils. If you put one of these tiny threads under an electron microscope, you see it has alternating light and dark bands.
And the notes say this all comes down to two key proteins. This is the dynamic duo of movement.
This is it, actin and myosin.
Let's distinguish them because I always get them mixed up. Which is which?
Okay. The light bands, the paler stripes, are called I bands. They're made up of the actin filaments. The key thing to remember is that actin is thin. It's a very thin protein filament, so light passes through it more easily.
I band. Isotropic.
Precisely. They are isotropic to polarized light, hence the I.
Light band. Thin actin. Got it.
Then you have the dark bands. They're called I bands. They are made of the myosin filaments. And myosin is a thick, bulky, complex protein. It blocks the light.
So it's anisotropic, hence the A.
You got it. And where the thick myosin filaments overlap with the thin actin filaments.
That's the darkest part of all.
It's the darkest part of the A band. And that zone of overlap is where all the magic happens. The entire trick of muscle contraction is happening right there.
Before we get to the magic trick, there's one more piece of the architecture here. The hook. The cross bridges.
Right. This is crucial. So visualize that thick myosin filament. Picture a thick braided rope. But sticking out from this rope, spiraling around it in every direction, are these little arms with heads on them.
The little arms.
Tiny little projections. We call them cross bridges. They stick out from the surface of the myosin, and they are literally reaching towards the actin filaments that surround them. These are the engines. These are the parts that are actually going to do the pulling.
So the actin is just the rope that's being pulled on.
Basically, yes. In this analogy, the actin is the passive track, or the rope. The myosin and its cross bridges are the active motor that moves along the track.
Okay. I have a mental image now. I've got thick ropes with arms, surrounded by thin ropes waiting to be grabbed. But what holds this all in a neat repeating pattern? Why doesn't it just turn into a tangled soup of protein inside the cell?
The Z-disc.
The Z-disc. This sounds like something out of a sci-fi movie.
It kind of looks like it. Imagine a scaffolding wall, a rigid plate made of protein that goes all the way across the myofibril. The ends of the thin actin filaments are bolted directly into this Z-disc.
Okay, so it's an anchor point.
It's the anchor point, and the Z-disc passes all the way across the myofibril, actually connecting to the myofibrils next to it, holding the entire cell in alignment. The space between two Z-discs, that specific repeating section of muscle machinery, is called the sarcomere.
That's the sarcomere. Okay, the notes bolded this term like 5 times. This is important.
It is the fundamental unit of contraction. It's the smallest possible piece of a muscle that can still perform the function of contraction. If you understand how one single sarcomere works, you understand how an Olympic weightlifter can deadlift 1,000 pounds. It's just billions and billions of sarcomeres working in unison.
There was a very specific number mentioned here regarding the length of the sarcomere, about 2 micrometers.
Ah, yes, the sweet spot, the optimal resting length.
Why is that specific length so important?
Think about it mechanically. You have these myosin arms, the cross bridges, reaching out to grab onto the actin. If the sarcomere is stretched out too far, say longer than 2.2 micrometers, the actin filaments are pulled so far away from the center that their ends no longer overlap with the myosin heads.
Because you're just grasping at air. The arms can't reach the rope.
Exactly. You get zero force generation.
And what if it's too short? Compressed too much?
Well, if it's squished down much shorter than 2 micrometers, two bad things happen. First, the actin filaments from opposite sides start to overlap each other in the middle, which gets in the way. And second, the ends of the thick myosin filaments literally jam up against the Z discs. The whole machine gets clogged.
So nature, through evolution, has optimized the bodies to basically live in this 2 micrometer window.
Precisely. The way our muscles attach to our skeleton, the length of our tendons, it's all designed so that when your arm is in a natural, relaxed, or slightly flexed position, most of your sarcomeres are sitting right at that 2 to 2.2 micrometer length, ready to generate the maximum possible tension.
That is just incredibly cool. It's like this suspension on a race car being perfectly tuned for the specific weight of the car and the conditions of the track.
It's exactly that. But there is one more piece of this architecture we absolutely have to mention, the unsung hero. The notes call it filamentous and springy.
Titan. Titan.
This sounded like a Greek god or something when I read it.
It might as well be. It is one of the largest protein molecules known to exist in the body.
Really?
Oh, by far. It has a molecular weight of about 3 million. For context, most of the proteins in your body are in the tens of thousands, maybe a couple 100,000. This thing is an absolute leviathan.
And what on earth is this giant doing in there?
It's the suspension system. It's the framework. Remember, the thick myosin filament is just floating there in the middle of the sarcomere. It needs to be able to slide, but it can't just float away or get off center.
So Titan is the tether that holds it in place. It's more than a tether. It's a molecular spring. One end of the Titan molecule attaches to the Z disc, and the other end attaches directly to the myosin filament. It acts like a bungee cord, holding the myosin perfectly in the center of the sarcomere.
And it stretches.
It's incredibly elastic. When you stretch your muscle passively, Titan stretches out. When you relax, its springiness helps pull the sarcomere back to its resting length. It keeps the entire intricate machinery perfectly aligned. Without Titan, even one strong contraction would probably scramble your muscle fibers into a disorganized mess.
It is the shock absorber and the centering spring, all in one.
Exactly. So that's the car. We've built the chassis, which is the sarcolemma. We've got the engine block, the myofibrils. We've got the pistons and the tracks, actin and myosin. And we have the suspension and framework, Titan.
But a car doesn't move just because you've built it.
Yeah.
You have to put the key in the ignition and turn it.
And that brings us perfectly to section 2, the spark.
This is the part that always feels like magic to me. I have a thought in my head. Pick up coffee. That is a brain signal. How does that signal, which is fundamentally electricity, cross the gap from nerve to muscle and turn into a mechanical pull?
It's an incredibly fast and elegant chain reaction. The source material outlines a clear 6-step sequence.
Okay, let's walk through them. Step 1, the signal.
An action potential, that's the electrical spike, travels down your spinal cord, out through a motor nerve, and races toward the specific muscle fiber it's supposed to control.
But it doesn't actually touch the fiber, does it? There's a gap.
There's always a gap. It's called the synaptic cleft, or the neuromuscular junction. The end of the nerve gets incredibly close to the muscle fiber, but they never touch.
So the electricity, the action potential, basically hits a dead end.
A cliff.
It hits a cliff. And electricity cannot jump that gap. So to get the signal across, the body has to switch communication methods. It has to convert the signal from electrical to chemical.
Step 2, chemical handshake. Right. At the very tip of that nerve ending, there are thousands of tiny little bubbles or vesicles, and they're filled with a special neurotransmitter called acetylcholine.
Acetylcholine, I've heard of that.
When that electrical spike arrives at the cliff, it triggers voltage-gated calcium channels to open. Calcium flows into the nerve terminal, and that forces these little vesicles to merge with the nerve membrane and burst. They spray their acetylcholine contents out into the synaptic cleft.
So it's not a bridge, it's more like a chemical messenger service.
It's a courier. The acetylcholine molecules swim across that tiny gap and land on the other side, on the muscle fiber membrane.
And that leads to step 3, opening the gates.
On the muscle membrane, the sarcolemma, there are specific protein channels. Think of them as baits that are locked. And acetylcholine is the specific key for those locks.
Okay.
When acetylcholine binds to these receptors, the gates fly open.
And once the gates are open, that's step 4, the rush.
This is the violent part of the process. The fluid outside the muscle cell is teeming with positively charged sodium ions. They are kept out by the membrane, creating a huge electrochemical gradient. They are desperate to get inside.
And the acetylcholine just opened the door for them.
It flung the door wide open. As soon as those gates open, sodium ions flood into the muscle cell.
It's an invasion.
A massive, instantaneous invasion. And because each sodium ion carries a positive charge, this flood of positivity dramatically changes the electrical voltage on the inside of the muscle membrane. It causes a depolarization.
So in that instant, we've turned the chemical signal back into an electrical one.
Exactly. We've successfully passed the baton. We went electrical in the nerve to chemical with acetylcholine, and now back to electrical in the muscle.
That's almost inefficient. Why not just wire the nerve directly to the muscle? Why the chemical middleman?
Control and modulation. A chemical synapse allows for much finer control. You can have inhibitory signals, you can modulate the strength. A direct electrical wire would be like an on-off switch with no dimmer. It would be far too twitchy and prone to misfiring.
Fair enough. So now the muscle has its own electrical signal, its own action potential. Step 5, the transmission.
That new electrical spike doesn't just stay at the synapse. It races along the entire surface of the muscle membrane, the sarcolemma. It spreads out across the fiber like a wildfire.
But wait, here's a problem. The actin and myosin, the actual machinery we talked about, are deep inside the cell. The electricity is just on the skin. How do we get the signal from the surface deep into the factory?
An ingenious bit of plumbing, the T-tubules.
The T-tubule.
Transverse tubules. The muscle membrane, the sarcolemma, actually has these deep tunnels that dive from the surface deep into the center of the fiber, weaving their way around all the myofibrils. The electricity travels down these tunnels.
So it carries the signal to the interior. And what is it looking for down there?
It's looking for a structure called the sarcoplasmic reticulum.
Step 6, the trigger.
The sarcoplasmic reticulum. It sounds like a villain's lair from a comic book, but it's actually an intricate, specialized storage tank. It's a web-like network that wraps around each and every myofibril like a lace stocking.
And what is it storing in this tank?
Calcium.
Just calcium, the stuff that's in milk.
Huge concentrations of calcium ions. Its main job is to actively pump calcium out of the cell fluid, the sarcoplasm, and hoard it, lock it away inside its tanks.
But why? Why go to all that trouble?
Because calcium is the master switch. It's the trigger. In the simplest terms, if calcium is loose and floating around the myofibrils, the muscle contracts. If the calcium is locked away inside the sarcoplasmic reticulum, the muscle relaxes. It is the final, non-negotiable on switch.
So when the electricity traveling down the T-tubules hits this tank,
it triggers voltage-sensitive proteins in the T-tubule that are physically linked to calcium release channels on the sarcoplasmic reticulum. It mechanically pulls open the floodgates. The sarcoplasmic reticulum dumps its entire calcium payload directly onto the myofibrils.
and that is the moment of truth.
That is the moment. The calcium hits the proteins, and the sliding, the contraction, begins.
It's just amazing to think that every single time I so much as twitch my eyelid, my body has to run this complex chemical baton pass. Nerve, acetylcholine, sodium, electricity, calcium.
And it does it in a matter of milliseconds. It's faster than you can think.
But here's the crucial follow-up question. If dumping calcium starts the contraction, how do we stop it? Otherwise, that'd just be in one permanent cramp.
You have to put the genie back in the bottle, and you have to do it fast.
So cleanup phase.
Exactly. As soon as the release happens, powerful calcium pumps on the membrane of the sarcoplasmic reticulum start working furiously to suck all that calcium back in.
And this costs energy. I
Yeah.
saw that in the notes. This costs ATP.
A lot of it. This is a critical point for everyone to understand. It takes energy, ATP, to power the contraction, yes, but it also takes a significant amount of energy to power the relaxation. You have to burn fuel to pump that calcium away and reset the entire system for the next signal.
which explains so much about what happens when you run out of energy. The
Yeah.
system fails.
It fails spectacularly. But we'll get to the grim details of rigor mortis later.
Right. Okay, so the calcium is out. It's been dumped. It's floating around the actin and myosin. Now we can finally get to the actual mechanics. Section 3, the molecular machinery in high definition.
This is where we need to put on our molecular goggles and really visualize the machinery. We're looking at what's called the sliding filament mechanism.
The source material makes a really important distinction here. It says the muscle doesn't shorten like an accordion or a spring.
Right. An accordion crumples up, the material itself gets shorter. That's not what happens in a muscle. The proteins themselves, the actin and myosin filaments, they don't change length at all. They stay the exact same length.
So how does the muscle get shorter if none of the parts get shorter? That seems like a paradox.
Telescoping. Think of an old-fashioned spyglass or a telescope collapsing. The outer tubes slide over the inner tubes. In the muscle, the thin actin filaments slide inward, deeper in between the thick myosin filaments.
Okay, so the zone of overlap increases. Let's look at the motor that actually does this sliding, the myosin molecule.
The beast.
The source describes this myosin molecule in incredible detail. It's not just a simple stick. It's a complex braid of proteins.
It's a massive protein complex. Each individual myosin molecule is made of 7 polypeptide chains, 2 very large heavy chains, and 4 smaller light chains.
The 2 heavy chains wrap around each other to form a double helix. That makes up the long, straight tail of the molecule.
Right, and at one end, the 2 heavy chains fold outward, each one forming a globular polypeptide structure. That's the head.
So it looks sort of like a two-headed snake.
A 2-headed snake with a very long, twisted tail. And here's the absolutely crucial part. Each of those 2 heads functions as an ATPase enzyme.
ATPase, meaning it has the ability to break down ATP.
It has the intrinsic, built-in capability to cleave a molecule of ATP and harness the energy that's released from that high-energy phosphate bond. That head is the engine. It's the part that burns the fuel and does the work.
And these individual myosin molecules then bundle together to make the thick filament we see in the sarcomere.
About 200 or more of them bundle together. The tails all point toward the center, forming the body of the filament, and the heads hang off the sides.
And the source notes that the heads are arranged in a spiral around the filament, covering 360 degrees.
So that it can grab in any direction. It's an omnidirectional pulling machine. It doesn't matter where the surrounding actin filaments are. There's always a myosin head nearby, oriented correctly and ready to grab it.
Okay, that's the motor. Now let's look at the track it runs on, the actin filament.
The actin filament is a bit more complex than just a simple strand. Its backbone is a double-stranded F-actin helix. The easiest way to picture it is to imagine 2 strings of pearls twisted together.
And on these strands of pearls, there are special spots, active sites.
Yes. On each of the little pearl molecules, the G-actin, there's an active site. These are spaced about every 2.7 nanometers along the filament. You can think of these active sites as the handles that the myosin heads are desperately trying to grab onto.
But they can't just grab them whenever they want, because if they could, I'd be in a state of permanent, agonizing cramp.
Exactly. There has to be a safety mechanism, a lock that keeps the myosin from grabbing the actin when the muscle is supposed to be relaxed.
Enter the regulatory proteins. First up, tropomyosin.
Tropomyosin. These are long, thin, thread-like proteins that wrap spirally around the sides of the actin helix. And in the resting state, the tropomyosin physically lies directly on top of those active sites.
Literally covers them up.
It's like putting child-proof safety caps on every electrical outlet. The myosin head is sitting right there, it's energized, it's ready to bind, but the handle is covered. Contraction is physically impossible.
So how do we take the safety caps off? How do we remove the tape?
We need a key. And that key is the third player in this trio, troponin.
Troponin. The notes describe it as a complex of 3 different protein subunits.
Think of troponin as the lock that's attached to the safety cap. It's a little complex of proteins that's intermittently attached along the sides of the tropomyosin. And its 3 subunits have very specific jobs, or affections, as the text puts it.
Okay, so troponin I has an affection for actin.
Right. That subunit helps bind the whole troponin-tropomyosin complex to the actin strand itself. It anchors it.
Troponin T has an affection for tropomyosin.
That subunit binds the complex tightly to the tropomyosin tape.
And then there's troponin C.
Troponin C has a strong affection, or binding affinity, for calcium ions. This is the ignition switch. This is the keyhole.
Ah, so here is where that flood of calcium from section 2 finally comes back into the story.
Precisely. Remember, the sarcoplasmic reticulum just dumped calcium everywhere. It's now bathing these filaments.
Which brings us to section 4, the walk-along theory of contraction.
Let's set the scene again. The muscle fiber is now flooded with calcium ions.
The calcium effect.
Those calcium ions immediately bind to troponin C. The text says that each troponin C can bind up to 4 calcium ions, and when it does, it's a very strong bond.
And this binding causes a shape shift, a physical change.
It causes what's called a conformational change. The entire troponin complex twists and changes its shape. And because it's physically attached to the
It
tropomyosin,
yanks on it.
it tugs the tropomyosin molecule deeper into the groove between the 2 actin strands. It physically pulls it out of the way.
It pulls the safety caps off the outlets, the tape comes off the handles.
It uncovers the active sites, and now, for the first time, the energized myosin heads have a clear, unobstructed target. The lock is open.
And this is the moment that initiates what the source calls the power stroke cycle, or, my favorite, the ratchet theory.
This cycle is the absolute heartbeat of all movement. It happens in 4 or maybe 5 distinct steps over and over again.
Okay, step 1 is preparation.
This is what's happening before the calcium even arrives. The myosin head is already cocked.
Cocked like a hammer on a gun.
Exactly like that. Or like a mousetrap that's been set. In a previous cycle, it grabbed an ATP molecule and cleaved it into ADP and a phosphate ion. It's still holding on to both of them, and the energy released from that split has been used to force the head into a high-energy extended position, pointing perpendicularly toward the actin filament. It's just waiting.
Step 2, binding.
The calcium arrives, the active site is uncovered. The myosin head, which has a natural chemical attraction to that site, immediately snaps onto it, forming a cross bridge.
Step 3, the power stroke.
This is the magic moment. The very act of binding to the actin causes a profound shape change in the myosin head. It tilts, it pivots toward the arm of the cross bridge, like a lever being pulled.
And it drags the actin filament along with it.
It drags the actin filament a tiny, tiny distance, about 5 to 10 nanometers. This tilt is the power stroke. This is the actual generation of force.
And as it tilts, it releases the ADP and the phosphate that it was holding onto.
Correct. You can think of it as spending that stored energy to perform the pull. So now the head is tilted, it's empty, and it's still rigidly attached to the actin.
Okay, now, step 4, release. And this is the part that is so counterintuitive, it always gets me.
It's the part everyone gets wrong. To let go of the actin, the myosin head needs new energy.
So wait, let me get this straight. It uses stored energy to pull, but it needs a fresh molecule of energy just to let go.
That's right. A new molecule of ATP must come along and bind to the myosin head. The binding of this new ATP is what causes the head to change its shape again and detach from the active site.
So if you run out of ATP, you literally can't let go.
You cannot let go. The muscle stays locked in a contracted state. That is the single most critical concept in this whole cycle. You need to spend an ATP just to buy your way out of the bond.
Wow. So the new ATP binds, the head detaches, then what?
That leads to the final step. Step 5, recocking. The head, now holding the new ATP, immediately functions as an enzyme again. It cleaves that ATP back into ADP and phosphate. The energy released is used to push the head back into its original perpendicular, high-energy cocked position.
And it's ready for the next cycle.
It's ready to grab the next active site down the line as long as calcium is still present. It's a ratchet. Grab, pull, release, extend. Grab, pull, release, extend.
And this happens billions of times a second across the entire muscle.
Billions upon billions. And remember, the source points out that each cross bridge works independently. It's not a synchronized swim team all pulling at once. It's more like a massive tug-of-war team where everyone is just pulling at their own rhythm. The total force you generate is the statistical sum of all those individual bridges pulling at that exact moment.
And this relates to something called the Fenn effect that was mentioned in the source material.
The Fenn effect is a simple but profound observation. It states that the more work a muscle performs, the more ATP it cleaves. It sounds obvious, but it means the system is demand-driven. If you need to pull a heavier load, you don't just pull harder, you actually cycle more cross bridges more frequently, and therefore you burn more ATP.
The engine automatically revs higher when the hill gets steeper.
A perfect analogy.
Let's shift gears a little and talk about the physics of this pull. Section 5, tension, length, and velocity. because this isn't just biochemistry, it's pure mechanics.
Right. And as we already touched on with the 2-micrometer rule, the physical arrangement of the parts dictates the power output.
The length tension curve. This graph in the notes is really telling. It looks like a bell curve.
It is. The peak of the curve, the maximum active tension, happens when the circumere length is between 2 and 2.2 micrometers. That's the point of optimal overlap between the actin filaments and the myosin cross bridges.
It's the point where the maximum number of hands can reach the rope.
Exactly. Now, if you stretch the muscle beyond that point, what happens?
The actin filaments get pulled away, the overlap decreases, and some of those myosin hands can no longer reach the actin.
Right, so the tension you can generate drops off. And if you stretch it far enough, there's no overlap at all, and the tension drops to 0. You can't pull a rope you can't reach.
Now, what about the other side of the curve? If the muscle gets too short?
If the muscle is compressed, the actin filaments from opposite ends of the sarcomere start to overlap each other, which interferes with the myosin binding. And even worse, the thick myosin filaments literally jam up against the Z discs.
The machine just bottoms out, gets clogged.
The tension drops off very rapidly. So this is why, for example, a bicep curl is hardest in the middle of the movement and feels weaker at the very top and very bottom. Your muscle is moving through this length tension curve.
Okay, that makes sense. Now, what about speed? The velocity of contraction.
There's a very specific and inverse relationship here between the load on a muscle and the speed at which it can contract.
It seems obvious, right? It's harder to lift a heavy box fast than a light one.
It is, but the physics behind it is precise. A muscle will contract at its maximum possible velocity when there is 0 load. Think of just flicking your wrist. As the load increases, the speed of contraction slows down progressively.
Why, exactly? Is it just friction?
It's the chemistry fighting the physics.
Yeah.
The load is exerting a reverse force that directly opposes the pulling force of the power strokes. If the load is light, the power strokes win easily and can cycle quickly. If the load is heavy, each power stroke is slower and more difficult. It's like trying to paddle a canoe against a strong current.
And if the load is heavy enough?
If the load equals the maximum force the muscle can generate, the velocity becomes 0.
An isometric contraction.
Exactly. You're pushing against a wall with all your might, but the wall isn't moving. The cross bridges are furiously cycling, they're generating massive tension, they are burning a ton of ATP, but the actin filaments aren't actually sliding anywhere. You're burning gas, but the car isn't moving.
And speaking of burning gas,
Yeah.
that's a perfect transition to Section 6, energetics.
Fueling the engine. This is so critical because, as we've established, ATP is the only currency the muscle accepts for contraction, for relaxation, for everything. But here's the problem. The amount of ready-to-use ATP stored in the muscle fiber is tiny.
How tiny are we talking?
The concentration is about 4 millimolar. That is only enough fuel to maintain a full maximal contraction for maybe 1 to 2 seconds. At most.
1 to 2 seconds. So if I decide to sprint for a bus and I only rely on the ATP currently sitting in my cells, I would seize up and faceplant after about 2 steps.
You would be completely out of gas instantly. It's clear that you can't rely on the pre-existing stores. You need backup generators. The body has 3 distinct and overlapping energy systems designed to reconstitute ATP on the fly.
Okay, let's go through these backup generators.
Yeah.
System 1, phosphocreatine.
This is the immediate high-power backup. Think of it like a supercapacitor or a battery booster pack. There's a chemical in the muscle called phosphocreatine, and it carries a high-energy phosphate bond that's actually even more energetic than the one in ATP.
So it can donate its energy.
Instantly. It can donate its phosphate group to an ADP molecule, recharging it back into a fresh ATP. The reaction is almost instantaneous.
That's your burst. But I assume the tank for this is also pretty small.
Very small. The combined energy from the stored ATP plus all the phosphocreatine can drive maximal muscle contraction for only about 5 to 8 seconds.
So that's the 100-meter dash. That's the single power lift. Pure explosive power, and then it's gone.
It's gone. So once that's drained, you have to fire up the next generator. System 2, glycolysis.
This is the breakdown of stored sugar, right?
Right.
Glycogen.
Exactly. The muscle cells store long chains of glucose called glycogen. Glycolysis is the process of rapidly breaking this glycogen down into pyruvic acid and lactic acid. This process liberates energy that's used to put ADP and phosphate back together into ATP.
What's the main advantage, the superpower of glycolysis?
Its superpower is speed and independence. It can form ADP molecules about 2.5 times faster than the oxidative system we'll talk about next, and crucially, it doesn't need oxygen to work.
So it's an anaerobic engine. This allows you to keep running even when you're out of breath and can't deliver enough oxygen to the muscles.
For a little while, yes. But it's a dirty engine. It's not sustainable. The accumulation of its end products, like lactic acid, limits this system to about 1 minute of full-throttle muscle activity. Think of a 400-meter sprint.
So we have 8 seconds of phosphocreatine, then about a minute of glycolysis. What if I want to run a marathon? Or just, walk around all day?
Then you need the big, clean, sustainable power plant. System 3, oxidative metabolism.
This is the engine for the long haul.
This is the system that provides over 95% of the energy for any sustained long-term contraction. It's the process of combining oxygen with the end products of glycolysis and with other cellular foodstuffs, carbohydrates, fats, and proteins to liberate massive amounts of ATP.
And for really, really long durations, like hours.
For endurance events lasting more than an hour, the body increasingly relies on breaking down stored fats. This is the diesel engine. It's slower to ramp up, but it runs for a very long time as long as you keep breathing and have fuel stores.
Now, the source brings up a somewhat, depressing statistic about muscle efficiency.
It is a humbling stat, isn't it? We think of our bodies as these high-performance biological machines. But the actual efficiency of muscle, meaning the percentage of energy input that gets converted into useful work output, is, at its maximum, less than 25%.
Less than 25%. That sounds terrible. Where does the other 75% plus go?
Heat. The vast majority of the energy you burn is lost as heat.
Which is exactly why you get hot when you exercise. It's not a side effect, it's the main product.
Exactly. It's the thermodynamic reality of the engine. We are essentially walking, talking heaters that occasionally move things.
I never thought about it that way before. I'm just a radiator with legs.
You are. But to be fair to our biology, that heat isn't entirely waste. It's vital for maintaining our core body temperature. But from a purely mechanical engineering standpoint, yes, it's inefficient.
Now, it's important to note that not all muscles and not all contractions are created equal. Section 7 breaks down the types of contraction and the different types of muscle fibers.
Let's distinguish the contraction types first, because we've mentioned them. Isometric versus isotonic.
Right. We touched on this. Isometric means same length.
The muscle generates tension, but it doesn't shorten, like standing in a doorway and pushing against the frame with all your might, or more commonly, just holding your posture. Your back and neck muscles are doing isometric work right now just to keep you sitting upright against gravity.
And isotonic means same tension.
The muscle shortens while keeping the tension on it constant. So, lifting a dumbbell at a steady pace, that's an isotonic contraction.
Okay, that's clear. Now let's talk about the hardware differences, the actual fiber types. Fast versus slow twitch.
This is where a lot of the gym bro science is actually rooted in real, solid physiology.
I feel like everyone has heard of fast twitch and slow twitch fibers. But let's break down the physiology. Why are they different? What makes one fast and one slow?
The text calls them type 1, which are the slow fibers, and type 2, which are the fast fibers, or even more descriptively, red muscle and white muscle.
Why are they different colors? Is this really like looking at a steak?
It is exactly like looking at a steak or a chicken. It's all about which fuel system they are optimized for. Slow fibers, type I, are built for endurance. Think of the soleus muscle in your lower leg that has to work for hours on end just to keep you standing.
What gives them that red color?
A protein called myoglobin. Myoglobin is an iron-containing protein that's very similar to hemoglobin in your blood. Its job is to bind to oxygen and store it inside the muscle fiber itself, creating a local reserve. It's bright red. That's what gives red muscle its distinctive color.
So red muscle is the oxidative engine. It runs on oxygen. It has its own oxygen tank. It can run forever, and it doesn't get tired easily.
Precisely. To support that, these fibers are generally smaller, they have a massive network of blood capillaries to deliver oxygen, and they are packed to the gills with mitochondria, which are the little power plants that perform oxidative metabolism.
Okay, now let's flip that and look at fast fibers, type 2, white muscle.
Think of the ocular muscles that have to flick your eyes around with incredible speed, or the gastrocnemius in your calf that you use for jumping and sprinting. These are built for raw power and speed.
And they're white because they lack that myoglobin.
Right. They have a deficit of red myoglobin. They don't rely on oxygen storage or oxidative metabolism. They are packed with glycolytic enzymes.
They use the dirty anaerobic engine.
They're the drag racer. Huge power, incredible acceleration, but terrible fuel economy, and they fatigue very quickly. To be fast, these fibers are physically larger for greater strength, and they have an incredibly extensive sarcoplasmic reticulum because they need to be able to dump and retrieve calcium at lightning speed.
So this is why a chicken breast is white meat.
Because a chicken uses its flight muscles, its pecs, for very short, powerful bursts of flapping to escape a predator. It's pure, fast-twitch fiber.
And the chicken leg and thigh are dark meat
The
because...
chicken stands and walks around on its legs all day long. That's an endurance activity. It's almost pure, slow-twitch, red muscle.
So my dinner plate is actually a perfect lesson in muscle physiology.
It always is.
Okay, Section 8. Control and mechanics. We have the engine, we have the fuel systems. How does the nervous system actually drive this incredibly complex car?
It does so by using what are called motor units.
What exactly is a motor unit?
A motor unit is defined as one single motor nerve fiber coming from the spinal cord and all of the individual muscle fibers that it connects to or innervates.
So one nerve connects to a bunch of muscle fibers.
It branches out at the end, yes. But the ratio of nerve to muscle fibers varies wildly depending on the job of that muscle.
The source makes a great contrast here between fine control and coarse control.
Think about the muscles that control your fingers for playing the piano or your larynx for singing an opera. You need incredibly precise, fine-grained control. In those muscles, the motor units are very small. One nerve fiber might control just 2 or 3 muscle fibers.
So you can adjust the tension by tiny, tiny amounts. You have very high resolution.
Precisely.
But then you have the coarse control muscles.
Like the big anti-gravity muscles in your leg, like the quadriceps, you don't need to twitch your quad by a single micrometer. You just need it to generate a lot of force to hold you up or make you jump. In those cases, a single nerve fiber might drive hundreds or even thousands of muscle fibers at once.
It's a much blunter instrument.
Effective, but blunt. And one interesting detail is that the muscle fibers within a single motor unit aren't all clumped together in one spot.
They aren't. I would have assumed they would be.
No, they're scattered throughout the muscle in little microbundles. This ensures that when a single motor unit fires, the contraction is weak, but distributed evenly across the whole muscle, rather than creating one localized, jerky knot of tension.
Okay, so here's the big question. How do we get stronger? If I need to lift a pencil versus lifting a piano, how does my brain signal that massive difference in force? It's all just an electrical signal, right?
The body uses a process called force summation, and it has 2 primary ways of summing up or increasing the force.
Okay, method 1, multiple fiber summation.
This relies on a brilliant management strategy by the nervous system called the size principle.
The size principle, how does that work?
It's very elegant. The smaller motor units, the ones with fewer muscle fibers and a smaller nerve, are more excitable. They're easier to turn on, so when your brain sends a weak signal to the muscle, only the smallest, weakest motor units fire first.
You bring in the interns first to do the light work.
Exactly, the interns. Then, as your brain decides it needs more force and increases the strength of the signal, it starts to recruit the larger motor units. It brings in the supervisors, then the department heads, then the VPs with the huge motor units.
And this allows for a smooth gradation of force.
Perfectly smooth. You don't want to use the force required to lift a piano just to pick a pencil off the table. You start small and add power incrementally as needed. It's what makes our movements fluid and controlled instead of jerky and spastic.
Okay, that's one way. What's method 2? Frequency summation.
This is about firing the same units, but firing them faster.
The text calls this tetanization. That sounds a little scary.
It does, but it's a normal process.
Yeah.
Imagine a single nerve impulse hits a muscle fiber. You get a single quick twitch that lasts a fraction of a second and then relaxes. If another impulse arrives after it's fully relaxed, you just get another identical twitch.
But if the next signal comes faster,
If the next signal arrives before the muscle has had a chance to fully relax, the second twitch builds on top of the first one. The force adds together, and if the signals come fast enough, so fast that there's no relaxation at all between them,
the individual twitches fuse together.
they fuse into one continuous, smooth, powerful contraction. That state is called tetanus, or tetanization.
So most of our normal voluntary movements are actually tetanic contractions, right? My arm doesn't vibrate when I pick something up.
Correct. The brain fires the motor neurons at a high enough frequency to create smooth, sustained motion.
There's a weird little phenomenon mentioned here called the staircase effect, or the German word Trepp.
Ah, yes, the warm-up effect. If you take a muscle that has been resting for a long time and you stimulate it with a series of twitches, the first contraction is actually relatively weak. The second one will be a little stronger. The third, stronger yet. It builds up over the first few contractions, looking like a staircase on the graph.
Why does that happen? Is the muscle just waking up?
Chemically, that's exactly what it's doing. The primary theory is that it's caused by a buildup of calcium ions in the sarcoplasm. With the first few contractions, the sarcoplasmic reticulum can't quite pump back all the calcium immediately, so there's a little bit of leftover calcium in the cytosol, which gets added to the new calcium released for the next twitch.
And more calcium means more active sites are uncovered on the actin.
Which means more cross bridges can form, which means more force is generated.
So the concept of warming up at the gym is physiologically real. You're literally priming the calcium pump and making the muscle more responsive.
It is absolutely demonstrably real.
Let's quickly touch on muscle tone.
Yeah.
Even when I'm just sitting here, my muscles aren't completely limp like a wet noodle.
No, they're not. Even at rest, there's a certain tautness that remains. This is what we call muscle tone. It's caused by a constant low rate of nerve impulses coming from the spinal cord. It keeps the muscles primed and ready for action and helps maintain posture and structural integrity. You can think of it as the engine constantly idling.
And finally in this section, the inevitable, fatigue.
Fatigue is, in many ways, the system protecting itself from damage. It has many causes, but one of the primary drivers is the depletion of the muscle's stored glycogen. The metabolic processes simply cannot keep up with the demand for ATP. The body essentially pulls the plug to prevent you from contracting yourself into a state of cellular damage.
Section 9, adaptation, remodeling the machine.
This is one of the most fascinating aspects of muscle. We tend to think of our bodies as being fixed structures. I have these muscles and this is what I've got. But muscles are incredibly plastic.
Plastic as in malleable.
Exactly. Malleable, changeable. They remodel themselves daily based on the functional demands you place on them.
Let's talk about the one everyone at the gym wants. Hypertrophy,
Yeah.
getting bigger.
Muscle growth. The source is very specific about the mechanism here. Hypertrophy is primarily an increase in the size of the existing muscle fibers, not an increase in the number of fibers.
Wait, so when I build muscle, I don't actually grow new muscle cells?
For the most part, no. There's a process called hyperplasia, the actual splitting and creation of new fibers, but it's believed to be very rare in humans, accounting for maybe a few % of the total size increase. What really happens is that the individual cells you were born with just get bigger.
How do they get bigger? What's happening inside?
They synthesize more actin and myosin filaments. The number of myofibrils within the cell increases, sometimes splitting. The machinery inside the factory becomes denser, larger, and more powerful.
And this whole process is triggered by near maximal load force.
Yes. You have to stress the system close to its limit to trigger the complex signaling pathways that tell the cell's nuclei, the current engine isn't big enough for the load we're being asked to move. Build more.
What about the opposite? Atrophy.
If you don't use it, you lose it. It's that simple. Disuse atrophy is the decay and degradation of those contractile proteins. The body is ruthlessly efficient. It will not pay the high metabolic cost to maintain muscle tissue that you aren't using regularly.
But there's a much worse kind of atrophy mentioned. Denervation atrophy.
This is what happens when the nerve supply to a muscle is cut. The muscle loses its trophic signals from the nerve and immediately begins to shrink rapidly. If that nerve connection isn't reestablished within a year or two, the muscle fiber itself is eventually destroyed and replaced by fibrous and fatty tissue.
And the text says this process is irreversible.
Once that muscle tissue turns into fat and scar tissue, a state called contracture, it cannot become muscle again. It's a stark reminder of how dependent the muscle is on the nervous system, not just for signals to contract, but for its very survival.
There's also this really cool adaptation regarding muscle length.
Yes, this blew my mind the first time I learned about it. If you immobilize a muscle in a stretched position, like putting a cast on a limb with the joint bent in a certain way, the muscle will actually add new sarcomeres onto the ends of the myofibrils.
It's like adding new links to a chain to make it longer.
Exactly. It remodels its own resting length to adapt to the new reality and prevent overstretching. And the opposite is true. If you immobilize it in a shortened position, it will remove sarcomeres from the ends to readjust. It is constantly tuning itself to the mechanical reality it lives in.
Okay, finally, Section 10, pathology. When systems fail, we have to discuss the grim reality of rigor mortis.
We do. It's the final and perhaps most illustrative lesson the muscle teaches us about its function.
It occurs a few hours after death. The body becomes stiff and rigid.
And why? Based on everything we've just discussed, you should be able to figure this one out.
Okay, let's trace it back. It all comes down to ATP.
Yeah.
You said that a fresh molecule ATP is needed to detach the myosin head from the actin.
Correct. That release step is an active, energy-requiring process.
Well, after death, breathing stops, so oxidative metabolism stops. The glycogen runs out, so glycolysis stops. The cell's reserves of ATP and phosphocreatine are depleted. There is no more new ATP being made.
And without ATP,
The cross bridges bind, they perform their power stroke, and then
they're stuck. They can't let go.
they are locked. The ratchet gets stuck in the pulled position. With billions of cross bridges locked onto the actin filaments, the muscles become rigid and unmovable.
And they just stay that way.
They stay that way until the proteins themselves start to break down and decompose due to autolysis, which is about 15 to 25 hours later, depending on the temperature.
That is horrifyingly logical. It is pure biochemistry. The system fails in the on position because the off switch requires energy. It's such a powerful reminder that what we call relaxing is actually an active, energy-intensive biological process.
Let's end with one more pathology, muscular dystrophy, specifically the Duchenne form.
This is a true genetic tragedy. It's an X-linked trait, so it almost exclusively affects males. The culprit is a mutation in the gene for a massive protein called dystrophin.
We mentioned the giant protein titan earlier. What does dystrophin do?
Dystrophin is another critical structural protein. We haven't talked about it yet, but it's the link between the internal machinery and the outside world. It connects the innermost actin filaments to the proteins embedded in the cell membrane, the sarcolemma. It essentially links the engine to the chassis.
So without it, what happens?
Without dystrophin, there's a critical mechanical disconnect. When the muscle contracts, the membrane is fragile and unstable. It tears easily. When it tears, calcium from outside the cell leaks in uncontrollably, which activates enzymes that literally digest and destroy the fiber from the inside out.
And the muscle just dies.
The muscle fiber dies and is progressively replaced by fat and scar tissue. It's a devastating progressive loss of the hardware itself. It shows that even one missing bolt in this incredibly complex machine can bring the entire system crashing down.
Wow. Okay, let's try to synthesize all of this. We have journeyed from an electrical thought to a chemical handshake, a calcium flood, the sliding of proteins, the ATP ratchet, the different fuel systems, and the long-term adaptations.
It is an immense amount of interconnected detail.
The thing that stands out to me after all this is the incredible fragility of it all. We think of ourselves as these powerful, robust beings, but it all hangs on this delicate microscopic balance of calcium concentrations and the constant availability of ATP.
That's one way to see it. I see it differently. I see the incredible robustness. Think about the elegance of the size principle for motor unit recruitment. Think about the built-in fail-safes like fatigue to prevent us from destroying ourselves. Think about the astonishing ability to remodel both strength and length on the fly. The system is profoundly adaptive.
That's true. I guess it's two sides of the same coin. Every time you lift a bag of groceries, your body is doing calculus at the speed of light to fire the exact right number of fibers, at the exact right frequency, fueling them with the exact right mix of sugar and oxygen from 3 different engines.
And it does it all completely unconsciously. You don't have to think about a single molecule. You just have the intent, lift the bag, and the entire magnificent machine obeys.
So here is my final thought for you, the listener. The next time you move, it doesn't matter if it's running for a bus or just tapping the pause button on this deep dive. Take a second to visualize those billions of tiny molecular ratchets in your muscles.
The myosin heads grabbing, tilting, and releasing.
The calcium flooding out of its tanks and then being furiously pumped back in. The ATP being burned by the trillion. You are a walking, talking, thinking marvel of biological engineering.
And whether it's 25% efficient or not, it's the only engine you've got. It's worth keeping it tuned.
Thanks for diving deep with us. We'll catch you on the next one.
Goodbye, everyone.